Hello, everyone. This is Senior Biotech Analyst John Vandermosten. Welcome to our channel that educates the life sciences investor on exciting advancements in drugs, biologics, and devices. For more content and news like this, subscribe to our channel below and like or share with others. Lexaria produced attractive results from its human pilot study evaluating GLP-1 agonists with its DehydraTECH platform. It compared blood levels of semaglutide from DehydraTECH formulated drug and Novo Nordisk's Rybelsus. So I wanted to ask the team at the company, which includes CEO Chris Bunka and President John Docherty, to tell us more about the study and what it found. John, I thought I'd ask you to speak with us first, since we hadn't had a chance to hear from you yet. Tell us about the human pilot study and what we saw.
Thank you, John. The study was a small investigator-initiated pilot study conducted in 7 healthy volunteers. The subjects were single-dosed with commercially available Rybelsus at 7 milligrams of semaglutide, and that was compared to DehydraTECH semaglutide, also at 7 milligrams in a capsule format. What we did is we looked at blood plasma levels of the semaglutide compared between the groups, as well as blood levels of glucose, as well as general tolerability. And we were really pleased with what we saw. We showed significantly higher levels in bloodstream of semaglutide, roughly 45% higher at peak. We showed significantly lower levels of blood glucose, and most notably that remained flat even after the volunteers consumed a standardized meal during the course of the study. And we saw lower incidences of unwanted gastric side effects. In fact, we saw 0 incidence of moderate nausea or diarrhea with DehydraTECH, whereas we did see those incidences with Rybelsus. So overall, it was a very exciting study, a pilot study that had some really promising results fueling our next steps.
Yeah, great. It sounds like that not only were the side effects better, but it had more durability, especially when the patients were having their meal. One thing you mentioned was that the DehydraTECH formulation used Rybelsus, crushed Rybelsus tablets. How might that affect the results? And do you think that that might be, the way you'd proceed in the future using that rather than semaglutide itself?
Yeah, that's a great question, John. For this particular study, yes, the DehydraTECH semaglutide capsules were prepared using commercially available Rybelsus tablets, crushing them as the semaglutide source input. Now, this was done so that the investigators had the highest quality semaglutide material from a patient safety perspective by levering the approved product. However, on the flip side, this also meant that the formulation included the formulation constituents of Rybelsus, such as their SNAC technology or sodium salcaprosate that powers their Rybelsus formulation. So moving forward through 2024, we have a packed R&D program where we're going to do some testing with and without the Rybelsus form of semaglutide in order to demonstrate and test if our DehydraTECH has the ability to work powerfully without the SNAC ingredient presence.
Right, and you're actually going to be looking at some of the other GLP-1 agonists that don't have the SNAC technology to rely on. So how will you combine DehydraTECH and those biologics to come up with something to give the patients in the trial?
Yeah, that's something we're very excited about. So we have the ability in, for instance, our upcoming rodent study work to work with semaglutide with and without presence of the SNAC technology. But we're also going to work with other actives that are not at all today formulated with the SNAC ingredient, like tirzepatide, which is commercially sold in the form Zepbound today as an injectable, and liraglutide, which is commercially sold in an injectable called Saxenda. So we have the ability to work with those actives sourced from those products without the SNAC ingredient present in a DehydraTECH formulation in order to test if DehydraTECH is effective without SNAC technology presence across a range of different GLP-1 and GIP active ingredients.
Okay. One of the things that I found very intriguing about the data that you shared in the human pilot study for the GLP-1 agonist was that when a meal was served to the subjects in the trial, the arm that had received the DehydraTECH formulation did not show the spike that was shown in the Rybelsus patients. And I was wondering if you could just share why you think this is, and what benefit does DehydraTECH provide that gives it that durability over time for that meal, so it doesn't spike the glucose.
Yeah, that was a very intriguing and exciting data finding from that first pilot study. Basically, the way that study was designed, all volunteers were fasted before dosing. It was a crossover study, so all volunteers experienced both test articles. And at the 4-hour mark after dosing, they were allowed a standardized meal. And interestingly, only in the Rybelsus treated group did we see a spike in blood glucose immediately following that standardized meal, whereas in the DehydraTECH semaglutide treated group, it remained flat. There was no spike. Reasons for the possible better control of blood glucose. First and foremost, we did deliver higher sustained levels of semaglutide than the Rybelsus achieved. So that may have had a key role in helping to attenuate blood glucose levels after the meal. But another very interesting facet of DehydraTECH is that it has been shown repeatedly in the past to enhance delivery of the active, not just into bloodstream, but also into brain. And we know, for instance, that one of the facets of GLP-1 drug effectiveness is through brain activity. So there is a possibility, and this is yet to be determined through more investigation, that part of the enhanced effectiveness in modulating blood glucose was due to enhanced brain delivery. One of our goals for 2024 is to test brain delivery of GLP-1 drugs in animals with DehydraTECH and hopefully see if that theory is true.
Now, will that be with the oral form of DehydraTECH, or because I know you have multiple formulations that can be administered? How might you apply that to get to the brain? I mean, I know that sometimes you can apply it orally, which may get it to the brain faster. Is that the case, or will you continue to use the standard DehydraTECH formulation?
Yeah, so for the large rodent study that we have planned for calendar 2024, the animals will be dosed through a means called intestinal gavage, which is to be delivered into the gut, and then it's absorbed from the gut into the bloodstream. So an oral, swallowable formulation. And the thing about DehydraTECH is that the nature of the DehydraTECH composition seems to be such that it allows the performance enhancement to not only be seen into bloodstream, but also from bloodstream and into brain. And we've shown this repeatedly in the past with other bioactive molecules in similar rodent studies. So for 2024, we want to test and see if that enhanced brain absorption is also occurring with things like semaglutide or liraglutide.
Great. Well, John, thank you so much. And again, we had John Docherty, Lexaria Bioscience's president, and also the company's CEO, Chris Bunka. Thank you.